Thank you, Dr. Nihar. You nicely pointed about the quality, and one of the quality measures is actually how good you are in responding to the environmental cues, and that's what we are talking about, the circadian rhythms, and in particular the liver clock with its implication in health and disease. So before I go into the outline, I just want to briefly mention the rationale why this topic is important. And this is a review article from Lancet Gastroenterology, which has pointed out very nicely the study done by Dr. Manoj in ILBS with almost 1,000 patients. And this study talks about the evening chronotypes and the sleep disorders in cirrhotic patients. So what exactly this means? That means the body is not adapting itself to the environmental changes which is taking place, and there is something called as a circadian misalignment. So with this background, I come to the outline. I'll divide this thing into two parts, the circadian rhythm, a little bit on the molecular clock, what all are the genes and what all are the rhythmic expressions. And in the second part, I'll just only talk about the liver clock a little bit with the ILBS data and what is there in the future. So when we talk about the definition, the circadian rhythm means something which is happening in a repeated behavior. And this repeated behavior, you as a clinician know it much better. This is the blood pressure, the heart rate, and many of the physiological events together with the growth hormone peaks. And how it is shown in the graph, when you have to draw it up, it's the peaks which come, goes down, and peaks which come, and it is spaced out either 24 hours in circadian rhythms. It can be ultra or infraradian if it is less than 12 hours or less than 8 hours. And what actually determines this rhythm? This rhythm is determined by a timekeeper, and the timekeeper is nothing but a molecular driver which is present in the central nervous system. And besides the central nervous system, which actually perceives the light, you also have non-photic cues like the food and the exercise. And why it is important again, biological clock is conserved in almost all the organisms in the planet, from the invertebrates to the vertebrates, and discovery of these molecular drivers ended up getting a Nobel Prize in medicine. So now if you have to discuss the rhythm and the clock, it's like an electric circuit which has three important things. One is an input. The input is actually dependent on how the earth is rotating around, and there is this constant 12-hour period of your lightness and darkness. This is actually perceived by certain pacemakers which have to adjust. So that means the body needs to adjust to these environmental cues. And that is taken care by the central nervous system in the suprachiasmatic nuclei, and these are mostly the photic cues. But what about the internal organs? They are not going to sense the sunlight. So what the internal organs does is, they actually synchronize with the brain. So brain is the master here, the CNS is the master, which actually, through the HPA axis, which is the hypothalamus, pituitary, and the adrenal axis, which also includes all the corticoids, and also many of the, so basically it's a neuroendocrine system. My knowledge is very little on that. So basically what the pacemaker is doing in the CNS is actually synchronizing all the organs so that they all can sense the time, what is happening outside. But even in an absence of this light, there is a non-photic cue. The non-photic cues includes the food and the physical activities, which can actually put you into a rhythm. So what exactly all this means is, that is the central, the peripheral, and each of these clock is actually built even in a cell. What they all are determined is of output. The output is actually the physiology and the behavior by which you kind of measure the quality of your life. That's one way to measure. So you have some rhythmic patterns which needs to take place, and if these rhythmic patterns are there, then actually the physiology is better. But many times there can be a delay in this, or it can be advanced in this, and that's where the problem starts. But the CNS and the peripheral organ make sure that they synchronize the whole thing. Now, what is its clinical relevance, or a medical relevance, I should say, when there is a desynchrony occurring? So what is inevitable is, with aging, there is a disruption in the rhythm, early morning pains as you age. But what is most important is the jet lag, which you all will feel when you travel to ASLD. So physically you will be in United States, but all your organs will be working as per the Indian Standard Time. So, but what happens is, this gets totally haywire or desynchronized in chronic conditions of, let's say, alcohol, or for that matter, you can say it lifestyle diseases. So now, from jet lag, there are two new terminologies. One is called as a social jet lag, but better scientifically is actually a metabolic jet lag. So what it is saying is, all these inbuilt clock system is maintaining the physiology through a metabolic output. So what this metabolic output is determined is by the pacemaker, and this pacemaker is just not present in the central nervous system, it's present in all the peripheral organs as well as also in the cells. So this timekeeper is actually nothing but a set of genes, and these set of genes very nicely sync in online with the sunrise and the sunset. So in the daytime, there are two important genes, which are the clock and the BML. These are transcriptional factors. They are active. When they are active, they rather transcribe. So these are transcriptional factors, so they transcribe a number of genes which actually go on accumulating, and by the start of with the sunset, their peak goes up. When their peak goes up, it actually shut down. So this is a transcriptional feedback loop. So there are genes which are active in the morning, there are genes which are active in the night, and this cycle goes on. And this is what was actually first discovered in Drosophila and later on found in the mouse, and now the same set of genes, the clock, BML, PER, CRY, all these genes are there in all the cells. And what is the output? The output is actually the clock control genes. So what are the clock control genes? Mostly those genes which are controlling the lipid metabolism, those which are controlling your glucose metabolism, those which are controlling your proliferation. So you can very nicely see that the metabolic clock is actually determined just by not by the CNS system, which is synchronizing, but also by the peripheral system. So with this background, you can see that the most important is, as the term now is the metabolic jet lag. So the dynamic duo of liver and the pancreas play a very important role in many of the chronic diseases. And I'm not going into all, but almost any organ that you take up, there's an inbuilt machinery of the BML and the clock, and accordingly, based on the light and the darkness, based on the feeding times, there are many things which are actually going on a circadian rhythm, and these could include the fatty acid, the glycolytic cycle, the lipogenesis and lipolysis, and so on and so forth. So with this background, I come to the second part, like what all are the components in the liver clock and how do you go about describing them. So these are very basic slides I have put, and very superficial you may think, but liver I think is a pulsatile organ. The reason being, there are many things which are having this typical circadian rhythm, going up at certain times of the day, coming down, then again going up and peaking. And what has been shown, so if you compile all this literature, you will find that 20% of the mRNA, and most of the pulsatile here, what we're talking about, is in the form of a gene expression. So the mRNA, 20% of them are oscillating. So if a human genome has close to 20,000 genes, so almost at one particular time, as I speak now, there are some 2,000 genes which are oscillating in each of these cells. The 20% of the soluble proteins, so this is your marker, which you see in the biochemistry, they are rhythmic, and that includes also the metabolites, which could be the lipids, your carbohydrates, and so on and so forth. So liver is one of the master clocks besides the SCN, together with the pancreas. I'm not touching the pancreas because I've read very little about that. So what all are the genes which are being controlled circadianly? These are the genes which are in the nutrients, like the AMPK, the mTOR pathway, and the mTOR pathway is one which you block even in a liver transplant. Then autophagy, many of the genes are there. Those in the glucose, especially the glucose transporters, the PGC1 alpha, and so on and so forth. But most importantly, from a fatty liver perspective, it is the lipid homeostasis. And this was a small work which we did, and you can see that there is a cyclic expression. So this is the cells are synchronized, this is an in vitro work, and we monitor it from 0 to 40 hours and take this, the supernatant and also the cell proteins at time intervals of at least 6 hours each. So you can find that there is some cyclical pattern which is there for PPAR alpha and a good cyclical pattern for SREBP. This is actually a lipid inducing, and PPAR is actually lipid, it helps in the lipolysis. So one of the most important things, and which was studied in the literature, was the glucose and the cholesterol metabolism together with the bile metabolism in the liver. So you can imagine as you wake up, so your eyes are open, your mouth is also open, you start feeding, and what goes into the intestine is taken up by the GLUT1 and the GLUT5, and the glucose is then uptaken by the GLUT2 into the hepatocytes. So this three important transporters, they have a cyclical rhythm. At least these are come from the mouse studies. So gluconeogenesis, glycolysis, and fatty acid metabolisms in mice peak at night. The GLUT1 and the GLUT5 peak at night. Why? Because mice is a nocturnal, and it is actually the activity which is determining and the food intake. Accordingly, the bile acids also peak late in the night, and the cholesterol biosynthesis. So these are from the mice studies. Same way what has been shown is for cholesterol synthesis, membrane channels, and most importantly, the detoxification system, which is your cytochrome P450. So this is all fine with the mouse, but what about the animal studies? And that's where there is actually limited studies available. The reason being, if you have to do a circadian rhythm, you need to take the sample for 24 to 48 hours at a timely interval of, let's say, 2 or 4, which becomes next to impossible. But there are very nice studies in the literature, and one which I found was in a gastro paper, where they talked about the bile acid and the cholesterol diurnal cycles, and how this is different actually from the mice. So you can see that at 1 p.m. and 9 p.m., the cholesterol peaks. So cholesterol is the blue one. So that means there are 2 peaks of these cholesterols coming. And what about the bile? Bile actually precedes it. Again, there are 2 peaks up. This is one at 12 a.m. and coming, and another at 6. So what it says is that although those diurnal rhythms are existing, but most of the studies being done in the mice may not be true for the humans. They may be having a different physiology. Now, I put this especially here because doing circadian rhythm is tough. In this particular study, in the gastro, there were only some 8 to 9 volunteers. Samples collected every 90 minutes. For an overall period of 24 hours. They were all put into a so-called metabolic and a sleep ward. The first sample was collected at 9 a.m. post-fasting, and then they collected all these data on the circadian rhythm. And the break and the food was actually very nicely time restricted with a good 6 hours of sleep. So this controlled environment becomes a little bit challenging when you have to do these kind of studies. It becomes easy in the mice. So what the mice study tells us, so if I had shown you the input and the pacemaker, so the pacemaker is actually the brain, and the brain is what synchronizes the liver. And what has been noted is, what if this pacemaker at the brain is taken away, will the liver be able to still sense it? And so there are some very nice, elegant experiments done in mice. So this is a normal mice, kept in a light and a dark cycle of 12 hours each, and followed for all the lipid synthesis and also for the cholesterol synthesis, and everything was perfectly in order as per the rhythm. Now, if you take out the brain, so you take out or delete, you make a knockout, and you make a knockout specifically only for the brain, then what happens? Will the liver is able to still sense the cue which is through the light, which is happening? So that means now the liver has to adjust to the environmental cues which are outside, but the brain is not perceiving it. And this elegant work, what it showed was that almost 10% of the mRNA and 20% of the metabolites are synchronized irrespective of the CNS. That way it says that it can work autonomously for certain things. So what these certain things are? These are the glycogen. So this is glycogen being made, and also glycogenolysis, and the glucose cycle, the NAD, which is a very important, like the ATP, this is also a kind of a currency, and the redox systems. So these are very important for the survival of the cells. So that means the liver can operate through its own internal BML, even in an absence of the brain central nervous system. But what if you delete this also? That's where the problem starts, and there is a the animals become lethargic, and there are a lot of metabolic derangements occurring. So this paper in Cell indicated that liver can detect changes in the environment, light, and capable of maintaining at least some basic functions independent from the brain clock, indicating about the autonomy. So now this is all fine in the mice. Now again we come back to the what is happening in the humans. So I'm going to show only 2 papers now. Both came in this year, and this is a work done by Sangeeta Bhatia. I think she's in MIT, and what it shows is that, so you think that the rhythm is only maintained in vivo. So that means as long as the brain is sensing the light. But let's say you take some part of your tissue from pancreas, or for that matter muscle or liver, and stick it up into a petri dish and put it into an incubator. They will still be doing the synchronized pattern. So what these people did was, and they have a good chips available, so they took the hepatocytes, the primary hepatocytes, put it into a dish, and then they synchronized it. And that's how we also do in the lab. We do a synchronization either by giving certain corticoids, or we mostly do it by what is called as a serum shock. And that's exactly what they did. And what they did was they looked at the circadian rhythm in these hepatocytes in vitro. And what they found was, this is a gene expression heat map. Without going to much of a detail, you can see some patterns. Some genes lighted up, same genes at a different time point they are going down, then again they are getting lighted up. So that means what? These genes are rhythmically expressed. So using this omic approach, they found that genes involved in detoxification, especially the cytochrome P450, again the same readouts of glucose pathways, glycogen, retinoic acid, and most importantly alcohol dehydrogenase and lipids, they all work in a cyclical way. So that means the cells try to maintain its cues for at least few hours or few days when you are kept it even outside the body. So what has this implication to do with for the clinical medicine? What these I'm sorry about this, it says circadian rhythms can influence drug effectiveness. So what they found was, one of the readouts which came was detoxification. Now if you see, the cells are synchronized, primary cultures. Now they're taking the cells after the synchronization, 24 hours, 30 hours, and they go up to 48 hours. And then they look for the expression of the genes. So you find that there is a cyclical expression of the cytochrome P453A, which goes up almost 1.5-fold to 2, and then it drops. Now they did a very nice elegant experiment in which they synchronized. So what cytochrome P450 doing? This is actually detoxifying the drug. So here they used 2 drugs which are relevant to the liver. One is acetaminophen. They also did the toxicity assays for atorvastatin, which I'm not talking about. So acetaminophen induces liver injury. So what they found is that the liver injury, it actually dependent on the time. So what do? So now if you give this acetaminophen at the time point 35 to 36 hours, post synchronization, as has been done, you will find that there is a 50% mortality occurring in the cells. So what that kind of indicates? This indicates that if somebody has to be given the acetaminophen, it's actually going to be deleterious or toxic. But if you can somehow manage or give the timely intervention in such a way that the dose and the time is matched, maybe you can prevent the toxicity. This is what the message is from this paper. One more thing which they did was, they found that the circadian control in the hepatocytes, it actually helps in the inflammatory response and infection. So that means what they are trying to say here is that the infection, when the virus goes and attacks, is also dependent on the circadian cue. So here what they did was, they did the experiment with Plasmodium falciparum. So again the cells were taken, they were synchronized, and then the Plasmodium was, this was given, the parasite infection was done. And again they found that if these cells are most receptive to this particular infection at the time point 36. So when they give at the time point 36, this is the time it responds, because at this time your inflammatory responses are down. That's why this parasite is persisting out. So what this implicates, that means the inflammatory signals are much stronger at certain times in the body, And does this mean that the viruses and parasites prefer a time of infection, and maybe they can be caught if you are giving a timely vaccine? So there are, I'm not going into the detail, but there are papers that for the influenza virus, the vaccination time, morning versus afternoon versus evening, makes a lot of difference. So this is what that the inflammation or the immune responses also work in a timely way. Now this is another very nice paper coming from Thomas Baumart's group in Nature Communication, and here they have done two things. So first, so in the previous paper, what they did was they just took the cells and plated into a culture dish. Now, as a clinician, you may not like that because you'll say that this is an artificial system. So what they've done a very carefully designed. What they did was they took the primary hepatocytes, they have a humanized mice. So now they take out the mouse cells from the liver and repopulate it with the cells from the human. And then they looked at all the genes which are circadian, because now this mice can be given the cues of light and day in a laboratory condition. And again the same genes popped up, those with bile acids, cholesterol, DNA repair, p53 signaling, and so on and so forth. And now into this experiment, they did another one. They put into this humanized mice the virus, which is the HCV, and they found that the cyclical disturbances in genes involved in the metabolism, genes related with fibrosis, gene related with the immunity. So what it says is that a virus can perturb this rhythmic function, and it actually takes help of this rhythmic function to actually help in its own proliferation inside the host. So this is all fine with respect to, so the only limitation which is there in the literature is all these studies which are done using the mouse model. So what is the output? Now output, we are looking at the physiological behavior. So Savera reviewed very nicely in her thesis what all are the genes which were knocked out. And this is, you don't have to read all that. The only crux up here is there are genes which when knocked out actually gives a change in the metabolic phenotype. And these metabolic phenotypes are mostly diabetes followed by the fatty liver. So you make a BML knockout, now you give a high fat diet, it will very nice fast progress towards a fatty liver phenotype. Also, if you look at the epidemiological studies, you will find that there are certain associations with certain germline mutations in the clock genes, and that gives rise to an evening kind of a chronotype. So most of these studies are from the animal for the circadian rhythm, knockout, mice kept in the dark, or they advance the light, so there's a phase change you can do. But the human association studies are mostly the genetic studies, and few studies which I have shown with few volunteers age. So I'm not presenting, but there's another paper which recently came up. They correlated the cyclic rhythm of albumin, melatonin together with cholesterol. And again, 8 volunteers, a paper in Nature. But these epidemiology studies have shown that the night shift worker and the metabolic jet lag, so there's a lot of information. So based on this, what WHO has done is they have classified the circadian rhythm as a probable carcinogen in the group 2A, making it a medically relevant topic. So what we did, quickly I'll come to the work, and what we in the lab trying to do was check whether there is association with the circadian rhythm in an in vitro condition. And the whole idea started when we found that there are some histone modifiers called the sirtuins. They prevent oxidative stress and apoptosis. Both these are actually hallmark features of NASH cirrhosis. And what we had actually shown, and many people had earlier shown, is that the sirtuin requires one particular cofactor, which is NAD. And the literature said that this NAD is in control of the clock genes. So we simply asked whether the clock genes can control the sirtuins and can it prevent the lipotoxicity. So this was a simple work done by a graduate student. So this is the cells which have been treated with saturated fatty acids, which is the palmitic acid, and it induced lipotoxicity at a dose of 600 micromole, and you can see that they do not regenerate, they do not proliferate, and they go into an apoptotic mode. Now in these cells, once the cells have been loaded with this palmitate, now you look at their circadian rhythm, you will find that in a normal cells you find a circadian rhythm of the clock genes. But this really gets disrupted when it is there when the cells are treated with this lipids. So we did a series of these genes, and of these, one of the readouts came out interesting was 32. You can see that there's a small peaks coming up in the normal. However, when treated with the lipid, there is actually a phase advance. So there's an acute peak coming up, and following that, it falls down. So this indicates a disturbance. So to cut the story short, what we found was this palmitic acid is actually causing a desynchrony of the two genes, BML and clock. And in turn, that lowers down the NAD. Because the NAD is lowered, we found that the SIRT2 expression and its function goes up. And what we uncovered was that BML regulates SIRT2 by binding to its promoter. I'm not showing that, but what will be the relevance? Now you see these pictures. These are cells loaded with palmitic acid, and the red droplets are the lipid droplets. Now, when you supplement NAD, what we find is now you cannot directly give the NAD to the cells. You have to give it in a form of a nutraceutical, which is a nicotinamide riboside, which actually increases the biological levels of NAD. You find that the NAD, this thing is still there, the lipids are still there, but to a lesser extent. But what if you block this SIRT2 by giving an inhibitor? This is how a ballooned hepatocyte looks like, nucleus pushed to the side, and this is a typical thing that you also see it in your biopsies, the balloon. So we could make some mechanistic links with this pathway. So now the question is this a potential drug target? And I'm just presenting one slide now. It's time for chronopharmacology and chrononutrition in NASH, and this is a concept. And here, right now, the two drugs which are being given are the obeticholic acid, and also there are preclinical trials of the FGF19. So here you can see that when this drug is given at 8 a.m. in the morning, there is a peak of this bile acids which is coming up around like 2 o'clock, and this is just after the lunch, and these are the placebo. This peak is the placebo. So in this concept paper given in JHAP, what the author describes is, instead of giving at 8 a.m. in the morning these two drugs, what if you combine and give it just before the meal time? You can actually lower this drug, and also you can avoid the toxicities. And so there is now, the question is, since many of these are clinical trials and there are prescribed timing, there are no prescribed timings as such. So if a prescribed timing can be given to these NASH drugs which are actually failing out in the clinics, but can the timing make these drugs more effective is the question. The other, I'm not covering much, but this is chrononutrition. This is for the nutrition department, and this is a nice study. So besides the light, the other thing which the nutrition time of restriction. So here they have done, this is a JAMA paper where they have looked at giving their food at a regular time between 8 to 4 p.m. and they find that the time restriction feeding reduces the hepatic, the fat content in the liver similar to that of a calorie restriction. So with that, I come to the summary. So I hope you appreciate about the BML and the clock, which are our master keepers. The genes which are under circadian controls are all those which you take for daily clinical parameters in biochemistry. These also includes the metabolites. A small work from ours and others people have shown that NAD could be a bit precursor. Now you cannot give a nicotinamide riboside as such, but it comes from the vitamin B3. So can something be done with that? And of course, you can impress upon your patients by talking about the social and the metabolic jet lags. But what I've not discussed is there's a lot of literature on regeneration and immune response, especially in the mice it says that liver regenerates at the night. Lot of paper and one recent paper which talks about the gut bacteria and its oscillation after FMT. And one big thing is the improving the skeletal circadian rhythm by exercise and nutrition rather than by the sun. And of course, in clinical therapies, consideration of the chemotherapy timings. But what is actually lacking is we need well-controlled circadian study, because right now the readout from a biochemistry is only one time point. So how to take into consideration for those genes which are important in the metabolism. and of course due consideration to be given in the clinical trials for the timing of the drug. So now I'll leave you with one small experiment that we are doing. So this, we're trying to develop a jet-like mice, and you can see that this is exercising very nice. This is a control one, but the one which had been actually phased advanced, it's not able to perform as good. Now using this, we can do a lot of studies with the circadian rhythm. So with that, I thank you and acknowledge. All the work was actually done here by Savera. She was a PhD student with Vipin, and all the clinical team who helped me collect the biopsies and the blood samples, and the funding agencies. Thank you so much.